Alright guys, it's 2:04, so I'm going to get started. So the topic of my lecture today is assessing individual differences with fMRI. And to start with, I like to just put up sort of examples of structural versus functional MRI, because I think even though those of us that work with bold data a lot sometimes forget just how stark the contrast is. So here we have a beautiful structural scan. These are used routinely, clinically all the time. And here we have functional MRI. So you can see already it would be very difficult for a clinician to look at a scan like this and extract any sort of meaningful information from it. The benefit of fMRI, of course, is that we get multiple time points, so we can observe, how brain function is changing over time. But at the end of the day, it's very difficult to extract any meaningful information from a scan like this that would give us some useful information in the clinic about what to do with any given patient. So really the way I see the overarching goal of looking at individual differences is to try to change this. So can we come up with ways of extracting meaningful and useful information from these types of scans that would help us out with individuals. Okay, so with that in mind, I'll give you a little outline of this particular talk. So first I'll talk a little bit about what we mean by individual differences. Then I'll go into a little bit of why we should care. I've already previewed that a little bit on the last slide. And then I'll move into sort of a more practical set of the talk of talking about how we can best study these. Hopefully many of you are already doing some kind of fMRI analysis and have your hands dirty with this, so try to give some practical recommendations. And finally I'll talk a little bit about the outstanding questions in the field and where we're going next. So to start with, what do we mean by individual differences? And so, as all of you probably know already, the way that fMRI is traditionally done is we give the subject a task in the scanner. We give this to multiple subjects, and each of them may actually have a very slightly different patterns of brain activity to the task, but typically what we would do is average these together in some way, because the traditional dogma is that it's very difficult to extract reliable information from any single subject. So this gives us sort of an overall map for what motor function looks like in a basic healthy human brain. And we can do this for lots of other types of tasks, and this has brought us, I would say, a decent amount of the way in terms of understanding the general blueprint for brain functional organization and what tends to be common across subjects. But along the way, I think many of us sort of lost sight of these individual differences, which is really where we want to try to get back to now. And so I'll actually be talking mostly today not about traditional task-based activation analyses, but more about functional connectivity. And unfortunately, we haven't had Katie's lecture yet. I think she's probably going to cover this very well on Monday, but just to give you a really brief overview of what this means, get us all on the same page. In functional connectivity, instead of looking at the magnitude of activation to a task in any single brain region, we're looking at pairs of brain regions or multiple brain regions at a time and how their activity is fluctuating together over the course of the scan. And so to make this more concrete, if we have a brain, we can divide it up into several regions, and then we can look at one of these regions, and let's say we look at that activity time course for that region over the course of our scan. If we then look at a second region, we can see how correlated or how, quote, functionally connected these two regions are. And in this case, they are fluctuating, their fluctuations are very similar, so we call that a fairly strong functional connection. And we can build out a matrix, so, oops, sorry guys, for each, basically each cell in this matrix is going to tell us how strongly correlated or how strongly functionally connected each pair of these nodes is in the brain. And so this is a nice way of analyzing data in the sense that it gives us a picture of whole brain organization. We don't necessarily have to choose a single region to look at. We don't even necessarily have to choose a task. So we can do this while people are doing tasks, but we can also do this while people are simply resting in the scanner, not doing anything explicit at all. And so, again, just a little bit of vocabulary. When I talk about these types of things, when we talk about brain networks, we often refer to the regions as nodes, and we refer to the connections between them as edges. So I'll use edges and connections sort of interchangeably. And this is an example of a typical parcellation that we might use. So these can take varying resolutions in terms of how many nodes we're using. This one happens to have 268 nodes. And one other point I'd like to make on this is that once you start looking at connectivity or interactions between brain regions, the number of variables you have scales exponentially. So with 268 nodes, for example, this gives us over 35,000 possible connections between regions. And this is both a blessing and a curse, especially when we're thinking about looking for meaningful individual differences. There's a lot of places that we could find them, but it's also harder to know which ones are truly robust and generalizable. Again, these functional connectivity analyses, which you'll hear a lot more about over the course of this summer course, have typically proceeded, in the early stages, people were doing them in very similar ways to traditional task-based paradigms. So they would scan a bunch of individuals and sort of look at sort of average or aggregate together their data in some way and find these general brain networks that tend to be present to some degree more or less in everybody and get this general blueprint for brain functional organization. But what we're hoping to do, again, is to sort of preserve these subtle individual differences in how these things are wired up or how they're connected in hopes that this will give us some traction looking into behavior or clinically meaningful biomarkers. Gosh, this is a struggle. Okay. So next I'm going to talk a little bit about, so how do we know, if we go back here, how do we know that these differences we're seeing are really meaningful and they're inherent to the individual and they're not just some sort of measurement error or some sort of other variability that we're not modeling appropriately? How do we know that they really mean something about each person? And so this was sort of my introduction to the field. So I'm going to walk you through this set of identification experiments that we did using data from the Human Connectome Project. So the idea here was that if these networks are stable in the same individual across 2 different days or 2 different scanning conditions, that would give us a hint that, again, this is something meaningful and something robust, something intrinsic. So the data set we're working with here comes from the Human Connectome Project. This was an earlier release of the data, so we had 126 healthy subjects, and interestingly, this data set contained 50 sets of twins, which only served to make our problem harder. And it was also a fairly homogeneous population, so it was all very healthy young adults, carefully recruited, etc. So if anything, there's probably less variability within this population than within many other types of fMRI studies. And so in the HCP paradigm, each subject came on 2 different days. So they would come on the first day, do a resting state scan, and also a working memory and a motor task scan. Then they would come back on a second day of scanning and do a second resting state scan as well as some additional cognitive tasks. And so what we did was for every subject we calculated one of those functional connectivity matrices like I introduced you to a couple slides ago. So we were able to calculate one of those for every subject for every task. And then we were asking ultimately the very simple question of can we match these connectivity profiles up between the same subject on 2 different days or 2 different scans. So can we pick someone out of a crowd based on their functional connectivity profile? So for example, we might try matching the 2 resting state scans, or we could even try matching between rest and a task or between 2 different tasks. So this is a pretty tough problem, but we'll see how we do. So the way that we did this was we would choose one condition to serve as the database. So for example, we might choose the resting state run from day 1. So we have all of our connectivity profiles calculated from this session. And then in an iterative process, so then we would choose a second condition to serve as the target set. So for example, rest from day 2. So then we go through this iterative process of taking a matrix from the target set. We don't know the identity of this matrix. We're trying to match it up to the correct subject in the database. And we do this simply by correlating this target matrix with each of the database matrices in turn and predicting the identity that has the highest correlation coefficient. So in this case, we might predict this comes from subject 2. Then we take a second matrix from the target set, go through and do the same thing. In this case, maybe we guess that it's subject 3, and in this way, if we go through the whole set, we can get an overall accuracy for how well we're able to do this. And that's what I'm showing here on this slide. So these are the results of that identification process, and here I'm just starting out with that example that I showed on the previous slide, so trying to match between resting states, so same condition but 2 different days. And it's important to point out that chance here is really, really low. So chance is roughly 1 in 126, since we have 126 subjects. And yet we observe that we're able to get the correct match about 93 to 94% of the time, which is really, really high, and that was exciting to us. And if we fill out the rest of these results matrices, you can see that if we're moving between rest and a task or between 2 different tasks, the accuracy drops slightly, but it's still well above chance. So by and large, the majority of the time, we're able to sort of pick you out of a crowd regardless of what you're doing while being scanned. Yeah?
Are the nodes that you're using the ones that you gave the example of earlier, and does the choice of nodes affect this a lot?
Good question. Yeah, so the question, sorry, I'm supposed to repeat the question because this is being recorded. So the question was about the choice of nodes and whether that matters. So yes, this is that 268 node parcellation that I showed a couple of slides ago. And I would say, so I'm actually going to get to that later, but my brief answer is that I don't think the exact parcellation boundaries matter. It's more about the resolution, so the number of nodes that you have. So if you use vastly fewer nodes, this accuracy will drop since you're sort of averaging over larger swaths of the cortex and whatnot. But I have a few slides on that coming up. Yeah, So this was ultimately, kind of a preliminary but still exciting result to suggest that there's some stability to these functional connectivity profiles that are present regardless of what people are doing and that have this strong individual component. But being neuroscientists, we wanted to know sort of where in the brain, which parts of the brain were contributing most to this identification result. And so to look at that, we took our 268 nodes and split them up into 8 different functional networks that you can see here. And for the next set of analyses, we were limiting identification to just the within network connections or the within network edges in each of these networks in turn to see if some of them would do better than others. And we found that indeed they did. So here what you're looking at is network number on the x-axis corresponding to these guys, and then identification accuracy on the y-axis. This is for the REST1 to REST2 pair, and these are for the rest of the REST to task and task to task pairs. And there's some variability, across the pairings, but overall, hopefully you can notice that the first 2 networks, so networks 1 and 2, which we're calling sort of medial frontal or frontoparietal networks, tended to do the best for identification. And this makes a lot of sense given that these are the networks in the higher order association cortices that people have previously observed have a lot of structural and functional variability across subjects. So that's what we may have predicted a priori, and that's also what we see, and that's also sort of further confirmation that there may be something cognitively or behaviorally meaningful about these differences. And just to sort of explore that point a little bit further, so this was a paper from several years ago now where they were also looking at individual differences in function, and they find that the biggest differences in these frontoparietal areas are also the ones that are the most evolutionarily recent, so the ones that have expanded the most since our non-human primates ancestors, and also that anatomical differences may be playing a role here. So these are also the areas that have the most variability in terms of sulcal depth and cortical thickness and things like this. So that's an important point, is that there are likely some anatomical differences that are playing a role here, but we think that there are some functional ones that are sort of going above and beyond that. And again, that's where we're really hoping to try to find some biomarkers. And this was another very recent paper. So since our paper came out, there's been some really additional really nice work and extensions and sort of replications and pushing this further. And this was just another really nice example from Katerina Gatan and the WashU group, where they're showing instead of having a large number of subjects here, they have only 10 subjects, but they've scanned them really, really densely. So each person has been scanned for tens and tens of hours doing rest and also doing different tasks. And they're showing basically the variance associated with each of the different factors. So you can see that at the group level, there's a lot of commonality. So this is just the variance explained in each of these parcels and the connectivity associated with each of these parcels by, for example, the group. And so there's a lot of shared organization, but you can see that once you take care of that, it's actually individuals that account for most of the variance here. So there's a lot more variance associated with individual, for example, than with the task that people are doing, which again recapitulates what we saw with the identification or the fingerprinting experiments. And there are some interesting interactions between individual and task. But overall, you can see that this factor here is really dominating above and beyond either task or session. So there really does seem to be something intrinsic about these data to each individual. Okay, so we've established that these individual differences exist, but why should we care about them? Again, I previewed this a little bit, but this is a point that I think is really, really important not to lose sight of. So I've already sort of alluded to this, but it's, you can imagine that it's fairly trivial to identify someone based on their structural scan. So this is just sort of a funny personal point, but when the fingerprinting paper came out, we were getting calls from news organizations saying, oh my goodness, this is crazy, we can identify people based on their fMRI, is the government going to use this against us, etc. And I just got such a kick out of it because there are way better ways to identify people, right? Like I can identify someone by looking at them. I can identify someone based on their DNA, based on their actual fingerprints. There are ways that are much more accurate and much more practical to identify people other than, putting them in an MRI scanner and extracting a functional connectivity profile. And then even just within MRI, it would also be trivial to identify someone based on a T1 weighted scan, for example. Which is expected to be very stable across days and definitely doesn't change, well, I shouldn't say that, doesn't change very much depending on what you're doing. Another criticism of this, it could just be anatomy, it could be other confounds, things like this. So how can we prove these differences are really meaningful? And, the gold standard for that for me is, do they relate to behavior? So do they relate in some meaningful way to sort of a real world output of the brain that we can also measure and relate to our fMRI. So we took an initial stab at this in the fingerprinting paper, where the HCP also provides a lot of out-of-scanner behavioral measurements on the subjects, and one of the interesting ones is this measure of fluid intelligence, which refers to the ability to discern patterns independent of learned knowledge. So this is a nice measure because it does vary a fair amount even among healthy adults, and so everyone in the database had excuse me, had completed this progressive matrices task, and so here was the distribution of scores in our sample of 126 subjects. And so we undertook really a fairly simple analysis where for every subject, we have their connectivity matrix. We leave one subject aside, crucially, to try to prevent ourselves from overfitting too much, but in our training set, we know their fluid intelligence score, and then basically we just search through the connectivity matrix to try to find edges or connections whose strength is related to performance on this fluid intelligence task across subjects. And then we can build a really simple linear model to relate these two things to each other, and then we can take in the connectivity information from this left out subject, pretend we don't know their fluid intelligence score, and try to predict it with our model. And when we do that, we assess this prediction by correlating the scores that we predict versus the scores that we actually observe. And we see that we're able to capture actually a decent amount of the variance in how people perform on this fluid intelligence test just based on their resting state functional connectivity, which again was exciting because it starts to suggest that these are meaningful, behaviorally relevant differences. And again, when we looked at the anatomy of which connections were doing the bulk of the predictive power, they were also in these same networks that were most discriminative of individuals, so in these sort of frontoparietal temporal association regions. And before and since and during then, there's been a lot of attempts to predict other behaviors, many of them successful. So we've also been able to show that resting state connectivity can predict people's ability to sustain attention. Dave Jangra in our lab has extended this to predicting reading ability, which I'm sure he'll talk more about in a couple weeks in this course. We've seen some initial traction that we can use this to predict symptom severity in psychiatric illnesses like ADHD and autism. And other groups have been extending this to try and predict personality traits like creativity, neuroticism, extroversion, narcissism, etc. So there's, I should emphasize there's still a long way to go. These predictions are far from perfectly accurate, but it's starting to suggest that we can capture meaningful variance in novel, previously unseen subjects in their behavioral measurements. And the last point that I'll make about this, I've talked very much so far about mostly healthy people, but I think that there's still really important relationships to eventual clinical utility here. So, and the idea here is to sort of move away from this more categorical approach where, say, people would typically, scan 20 controls and 20 patients with some neuropsychiatric illness, measure something about their brain, show a mean difference. So you can see, there's this statistically significant difference here, but these distributions are highly overlapping. And our intuition, the intuition of many others now, is that we may be better served by moving away from this towards a more dimensional approach where instead of stratifying based on diagnostic status, we're measuring a more continuous phenotype, be that performance on a task or some sort of symptom severity, something that's going to allow us to sort of put everyone on the same axis and take advantage of the full spectrum of variation in the human population. Again, with the ultimate goal of being able to bring in a brand new person, measure something about their brain, and then predict something about a continuous phenotype. Okay, so with that, I'm going to move into sort of a more practically minded section of the talk, talking about how we can study individual differences. So we still have a ways to go, and my colleagues and I have learned a lot over the years about ways to do this well and ways to not do it so well. So I'll try to give a few practical guidelines and the stuff that we've been thinking about. So one of the first questions that we got a lot after the identification paper came out was, do you need HCP quality data? So the HCP had very high spatial and temporal resolution, and people were wondering if that was really critical for this type of analysis, and my answer is not really. So it turns out that at least the identification part is fairly robust, even at more standard spatial and temporal resolutions. So this was a nice paper that came out a couple years ago by Iran et al. Sorry, this is a little bit small, but essentially they're comparing, these 4 graphs come from 4 different data sets that have different voxel sizes and TRs, and their metric actually is lower is better. So lower is a higher identification rate, and the x-axis here is just acquisition time. But you can see that even in these sort of more, I won't call them low quality, but more standard quality data sets, that the identification converges fairly quickly. So this is only about 10 minutes of data in the extreme, and you're still able to get pretty good results here. This is some really interesting work from Jason Drusgall down at UVA, where they're using HCP data, but they're artificially downsampling by a large factor the TR, or the temporal resolution. And so they find that even taking only about every 10th image, every 10th volume, and calculating connectivity matrices that way, they're still able to get a really good accuracy, and this only really starts to drop down after about 12 seconds. So it seems that this identification is fairly robust to those things. To get back to the question about parcellations, it's a really interesting one. So this Iran paper looked at that a little bit as well, and they found that the exact parcellation didn't matter too much, although more nodes tended to lead to a higher identification rate, which makes sense if you think about it. The finer grains resolution you have, sort of, the more you may be potentially capturing those meaningful individual differences, whereas if you're averaging over really broad parts of the cortex, you may be washing them out a little bit. But one caution with that is that in its extreme, the really high resolution parcellations may be amplifying the effects of anatomical differences or registration error. So we and other groups, it seems like we're sort of converging on this range from 200 to 300 nodes seems to be a decent compromise for that. But the question parcellation is still wide open, and there's a lot more work to do there. What about amount of data? So one thing that we've seen does matter a lot is scan duration, and this may not be surprising to many of you, but it's worth reiterating that longer acquisitions are always better. So this is bad news for the subjects, but to the extent that you can keep them in there longer, it's going to be better. So longer acquisitions give us higher reliability within subjects, seen here as interclass correlation coefficient as a function of scan length. So this is, again, within single subjects. We also observed this in our identification experiments, which are more of an across-subject measure, that the longer you scan, just the better characterization you're going to get of each individual. And one sort of interesting subtlety here is that a higher sampling rate, or in other words, a shorter TR, can't necessarily make up for a shorter scan duration. So this is another figure from that Iran paper. And you can see here, so these 4 lines are 4 different data sets with different TRs. And on the x-axis, they're holding constant the number of data points. But of course, the scans that have shorter TRs are going to reach that number quicker in terms of minutes in the scanner. And so this red line here is actually the data set with the shortest TR. I think it was around 600 milliseconds. And so this one takes the longest to converge. So you can't necessarily make up for a shorter scan duration by just sampling more quickly. It seems like the way that I like to think about this is there's going to be a certain number of states that the brain kind of traverses maybe, and the longer that you're scanning an individual, the better able you are to capture that variability within subject, which then will sort of average out when you start to do your cross-subject comparisons. But yeah.
I guess actually what you just said kind of is related to my question. So when I'm thinking about longer tasks or longer scanning, and maybe there's multiple things that are happening during the scan, I guess it kind of begs the question of this static versus dynamic connectivity. So have you looked, I don't know, just can you talk a little bit about
yeah.
the connectivity is stable over time within a person if different things might be happening?
For sure. It's a really good question. And I basically am not going to touch, I don't have slides on it because I haven't done as much with dynamics. I will tell you that the way that we got the original identification result was we actually were trying to get states. So we had all of the HCP conditions, and we had calculated, this was back in the day, so we were calculating sliding window matrices from the different states and the different subjects, and we threw them into a big clustering algorithm expecting to get out task or condition, and instead we kept getting subject. And so that was sort of our first clue. But that does not mean that there aren't dynamics within subject that are meaningful. I 100% believe that there are. I haven't done as much work with that, but certainly people have shown that dynamics add something above and beyond the static connectivity when you're trying to classify patients versus controls, for example. People are starting to, Oliver actually, who was in our lab last year, was doing some really nice work with Javier's data set looking at this, how dynamics, especially within task. I think dynamics within task are really, really interesting. And can you relate that to behavior on the task in the scanner? Certainly there's a lot going on there. Yeah. And then just to sort of hammer home that point a little bit more, this is a really interesting analysis where they're basically looking at the effect of truncating data or using less data, but they're doing it in two different ways. So here in the red line, they're scrubbing, so they're just randomly removing frames from any part of the run, whereas with the blue line, they're actually truncating contiguous frames from the end of the scan. So each of these is matched for amount of data, but it just seems like having samples from a longer period of time is really critical here. Okay, next question. Does scan condition matter? This is a question that's near and dear to my heart right now. I've been thinking a lot about this. So, one of the things that you could potentially take away from the identification results is that it doesn't really matter what people are doing in the scanner, right? If we're able to identify you, regardless of whether you're resting or doing working memory, doing language, or whatever you're doing, maybe scan condition doesn't really matter. And I think that's absolutely not true. So that was a lot of double negatives. So I think scan condition does matter a lot. It's something that we can use and we can exploit to try to pull out the individual differences that we most care about. And I like to sort of introduce this with kind of a thought experiment here, so bear with me. But let's imagine that we have 3 individuals that we'd like to study, and these individuals are correlated with one another at some ground truth average correlation of 0.5. In this case, this is the actual just correlation of the RGB values in these images with one another. But let's imagine that this is a latent variable that we can't observe directly, so we have to put these people into some kind of state in the scanner in order to measure their individual differences. And so we could think about states that make subjects look more or less similar to one another. So we could think about giving someone a task that sort of brings them all together and makes them look the same. This on its face might not seem like the best way to study individual differences since we sort of blurred them all out. So maybe what we want is a condition that makes them look really dissimilar to one another. But if we just sort of do this in an unprincipled way, we could end up, scrambling, so to speak, or essentially removing the meaningful variance that allows us to quickly identify people. So maybe the factor that matters more than just raw similarity across subjects is this metric that I'm calling identifiability. So we could think about this spectrum from more to less identifiable. And what I mean by that in this toy example is just how easy is it to look at these pictures and know exactly, know quickly who these people are. So we could think about a condition that makes people look less similar to one another, but in a way that enhances, their differences, and so quickly allows us to know who they are. And then, somewhat paradoxically, we could also think about a condition that overall is sort of bringing people together in this connectivity space, as I like to think about it, but in ways that are preserving or even enhancing sort of the most important subset of features that allow us to quickly look at these images and identify people. So I just put this up here as a way to start thinking about this type of space and where different scan conditions might fall within this space. And so I think this is a really important point, especially right now, because it seems like resting state has really become the default condition for both functional connectivity and individual differences in a lot of ways. Well, I talk to the task fMRI people, and they kind of say, yeah, duh, we've been using tasks to study individuals for a long time. But I think, especially with these larger data sets that are coming out and everyone sort of doing REST does have a lot of advantages. It's very easy to acquire, it's easy to standardize across sites, it's easy for populations that struggle with tasks, etc. But I think it's really worth, considering using tasks more deliberately because they may increase our signal to noise. So this was a follow-up experiment that we did replicating the identification experiments in a larger sample. This time we're also crucially controlling for scan duration, which we didn't do a great job of in the original paper. So these numbers are lower than what we had shown before. They're still well above chance. And this is a busy slide, but the thing that I like to point out here is that when we replicate these in the bigger sample, we're still seeing pretty good accuracy between the two REST scans. So we're getting about 70% when we go from REST1 to REST2. But interestingly, this is not as good as many of the task pairings. So even when we're going between two totally different tasks, so for example, relational to working memory, we get a higher identification rate. So people are doing different things, but it seems like just having any sort of task on board is serving to kind of stabilize the individual functional architecture in a way that allows us to identify people with more accuracy. That's the way I like to think about it. And there's also sort of an interesting result here whereby, thinking back to that space of those sort of orthogonal axes of similarity versus identifiability, when we correlate just the mean between subject correlation, so this x-axis here is just how similar on average are subjects' functional connectivity matrices in that condition. And we plot that against what I'm calling the mean database identification rate, which is just the mean across each of these rows. So how well on average does a condition perform as a database? We actually see a strong positive correlation between these two things. And so the easier way to think about that is that conditions that make subjects look more similar to one another actually make better databases for identification. And that was sort of a surprising result on its face. And you can see down here, the two resting state scans as well as the motor scan were scored lowest on between subject similarity. So these are the ones where subjects look most distinct from one another, which is what we would expect since we're not constraining what they're doing as much. This most challenging task here, the relational matching task, is the one that made subjects look most similar. But I say somewhat paradoxically, this one, these three also had the highest, performed the best for identification. And so it seems like even though we're kind of bringing everyone together, we're still preserving or even enhancing those most meaningful differences. And so if we bring back our theoretical space here, we can start to think about maybe these three tasks, which happen to be the emotional, gambling, and relational tasks, as maybe falling somewhere in this upper right quadrant of overall people are looking more similar, but the most important features that allow us to identify them are still there, and if anything, they're enhanced. Okay, so all of this, this is sort of a straw man argument. Is REST best for studying individual differences? I'm going to say probably not. Something that I and many of us now have been thinking about is using more naturalistic tasks. So all the tasks I've shown you so far come from the Human Connectome Project. They're very classic, sort of traditional psychological tasks, but there's a lot of interest currently in using more ecologically valid, or what we call naturalistic tasks. So this means just showing subjects a movie or playing them a story in the scanner, doing something rich and engaging. This is an example called Inscapes, which I think Dan alluded to a little bit on Monday. This is what has been called a glorified screensaver. It was actually developed initially as a compliance mechanism for young children. So if we're trying to scan kids, they don't do too well with REST. I mean, anyone who's been in a scanner, REST is pretty brutal, but kids especially don't do that well. And so the reasoning of Tammy Vanderwall, a collaborator of mine, was that, if we give them something pretty to look at, it's not going to drive the system too hard, but it's going to sort of occupy them, then maybe they'll stay still and we'll get better data. And she did, in fact, find that. But then we started wondering, what would these types of conditions do to our individual differences? Like, is it the case that by giving everyone the sort of same complex, engaging, continuous stimulus, are we going to wipe away all of that individual variability that we care about or not? And it was an empirical question. And so Tammy ran the experiment and found actually that identification rate during naturalistic tasks was just as good, if not better, than at REST. And so this was a data set of 34 healthy adults that were scanned on 2 different days in these 3 conditions. So REST, this Inscapes movie that I just showed you, and then also comparing that to a clip from Ocean's 11, so contrasting it with a sort of fast-paced, more truly naturalistic movie experience. And this is the identification matrix for those experiments, and you can see that the rates are pretty high across the board. Again, this is a smaller group of subjects, so chance is higher, but these are still well above chance. And if you look at the diagonals here, these are the identification rates between 2 matched conditions, so REST to REST, or Inscapes to Inscapes, Oceans to Oceans. And again, these are all really high, but if anything, the movie identifications are actually higher than the resting state ones, which is super interesting because, again, on the whole, you would imagine that subjects' brains are looking more similar during this movie, and that's true. But you still see these meaningful individual differences persist or even enhance. So that was really exciting to us because it suggests that we might be able to use these types of movies to constrain what the subjects are doing in a meaningful way and still be able to look at individual differences. Okay, I'm going to switch gears a little bit for the next few slides and talk about behavior, since, as I mentioned, I think that's really that's the gold standard here. So it's just individual differences in and of themselves are less exciting to me unless they relate to out-of-scanner behavior. And so one thing that's important to think about when you're choosing a behavior to study is to ask yourself if it's a stable thing within individuals. So this gets at the question of trait versus state. State variables are interesting, but they may be better to a within subject analysis. And there's a lot of interest in that right now. So some of you may have heard about this data set with a researcher named Russ Poldrack, who scanned himself, I think, 84 times over the course of a year and a half at REST and also doing different tasks. And he had a lot of state-related questionnaires that he would fill out on each of the scanning days. And people are starting to find some interesting correlations between brain connectivity measures and some of these state measures, like for example, surprise, or just generally how positive he was feeling that day. And so these are very interesting, but again, better suited to within subjects. So like something like surprise is less meaningful across subjects. Maybe this is obvious, but another thing to ask yourself is, does the behavior that you're interested in show a good distribution in your population? And this can be challenging sometimes, especially with the very healthy adults that we tend to get as healthy volunteers, here at NIH and other places. So for example, this is a distribution of performance scores on the emotion task in the HCP in-scanner performance. And you can see that there's a huge ceiling effect here, so everyone did super well on this task. It might not be that meaningful to try to look for individual differences that correlate with this because there's just not that much variation to begin with. However, something like the relational matching task, which was a more challenging task, did show a better distribution, and so this might be a better target for looking at individual differences. And again, maybe this is obvious, but I review papers all the time that indicate to me that it's not. And so just something to think about. In terms of mitigating confounds with your behavior, this is also a really big one. So this is probably not news to many of you, but there are many, many behaviors and phenotypes that are correlated with head motion in the scanner. Head motion, as you may or may not know, is a huge problem for functional connectivity analyses. I know Dan talked about this a little bit on Monday. But this is really a problem when you're looking at individual differences because you'll have these systematic variations in head motion that are scaling with exactly the behavior that you care about. So this was a study also using HCP data and just looking at correlations between the various out-of-scanner measurements and head motion in the scanner, and they're finding, again, pretty systematic differences that go in the direction you'd expect. So the subjects that are more compliant or had less head motion in the scanner also tended to do better on the out-of-scanner cognitive performance measures. Some of the things that correlated positively with head motion included things like BMI or other sort of physical health measures. And so this is something that's pretty pervasive, and there's ways of dealing with it, but it's important to check for these types of correlations. In general, patients of any kind are going to move more, young children are going to move more, and older adults are going to move more. So it's really important to keep this in mind. Ways of dealing with this, check for the correlation. You can't do anything about it if you don't look for it in the first place. You can consider excluding particularly high motion subjects. Again, this is sort of fairly obvious probably to many of you. One thing is to choose appropriate pre-processing techniques, which could be a whole talk in and of itself, but I thought this is a pretty interesting recent paper where each of these little boxes is the same exact data analyzed with different pre-processing streams, and they're looking to see how many of the edges in the brain correlate with head motion after different pre-processing strategies. And so you can see that, if you do basically nothing, almost all of the brain is going to be correlated with head motion, whereas you can really reduce those systematic correlations if you do careful pre-processing. You can also, of course, always use motion as an explicit covariate, which I would always recommend doing when you're looking at individual differences. Another plug for naturalistic tasks, I think Dan also mentioned this, but these tasks do decrease head motion in both children and adults. Actually, there's some evidence that any task will do this over and above rest, as long as it's not like a really miserable task that people just can't do. But even just anything to do while they're in there will help them comply, and people just tend to really like movies, so that can help you out. What is the best brain state? This is a question that I ask myself, and I also get asked all the time, and obviously we don't know yet, but I have the intuition that it probably depends on the behavior that you are trying to study. So we've seen that certain task conditions actually generate better predictions of behavior, and this is some work that Natasha in our lab has been extending and really nicely. This is an older figure now, but basically this was analysis we did, turning back again to the HCP data, where we're looking at if we build those predictive models that I mentioned briefly several slides ago. So if we're trying to build a model based on functional connectivity and then use that to predict a new subject's behavioral measure, we can vary the input that we give to that model. So we can give it a matrix from any of these conditions, from resting state, from working memory, from the motor task, and we can use that to try and predict various different behaviors. So here, this is a matrix of how well we're able to predict certain behaviors based on certain input data or certain scan conditions. And so here we have the fluid intelligence measure that I've talked about. This is a measure of vocabulary, and this list sort is a working memory task. And the takeaway that I just want to point out here is that the rest scans actually did the worst. These are still significant, so they're above chance, But the exact same subjects, trying to predict the exact same behavior, the model works much better if you just feed it the working memory data. So even to predict something that's not necessarily related to working memory, like this vocabulary measure, we're better able to predict vocabulary while people are doing a working memory task than while they're just resting. And I think this is a really compelling point. And one interesting thing to look at is if we look at our top 2 performing conditions for predicting any of these 3 behaviors that we've looked at here, they were the working memory and the language scan. And so if we bring back our space here in terms of similarity versus identifiability, we see that these are actually sort of somewhere in the middle. So they're associated with sort of average between subject similarity and average identifiability. And so if we bring back this theoretical space here, they might sort of fall somewhere up here or even somewhere more in the middle. And so this is just the types of things that we're thinking about when we decide, what conditions we might want to scan people in order to predict certain behaviors. Actually going back to that really quickly. So one idea that we're playing around with now, which I don't have a slide for yet because we don't have data yet, but the idea is that can we sort of design a bespoke scan condition to pull out variation in behaviors that we care about. So for example, if we want to study anxiety, is it better to have people watching an anxiety-inducing movie? Or if we want to study depression, is it better to have people watching a movie with social themes or things like this versus a neutral movie or a positive movie or things, basically how can we best maximize our ability to pull out the variation related to the traits that we care about by manipulating what people are doing in the scanner, since we've seen that it does matter. And so can we control this directly and design something that's going to give us the best predictive power, and that's something we're really interested in right now, so stay tuned. Okay, we are getting there. I just have a few more slides based on our instructions for this course, which was to talk about the outstanding challenges and where we're going next, and there's many of them, and hopefully they've become clear over the course of the talk, but I just want to spend a little time talking about this. So I see the outstanding questions as sort of falling into 3 themes. So there's data acquisition, there's data analysis, and then there's interpretation and applications. So some things that we need to be thinking about on the data acquisition side, this question of population. So who do we want to scan and how many people do we want to scan? I think this is a big one these days. So is it better to have many individuals lightly sampled or a few individuals densely sampled? Ideally we would have many individuals densely sampled, but with finite resources, is it better to, scan just a few people many, many, many times so we can make sure what we're seeing is really reliable at the single subject level, or is it better to sort of pull from a wider swath of the population? My bias being interested in behavior is that it's, you do need a certain distribution of scores on your behavior in order to meaningfully look at that. But I think there's been some really nice studies of just, dense phenotyping of one or a handful of individuals that are also teaching us a lot. And so eventually, hopefully we can sort of merge what we're learning from both of these types of studies to better leverage these types of data sets. I've talked a lot about healthy controls since that's the data that's easiest to access, but extending all of these approaches to patient populations is obviously going to be a huge thing, and people are already working on it. Don't mean to imply that they're not, but I think sometimes these larger data sets are capturing, a certain slice of the population that tends to come in for these types of scans. And if we're gonna, put our money where our mouth is and make translational tools, then we're gonna have to make sure that what we're finding is gonna work, in a patient population that's gonna be more challenging to get high quality data from. Longitudinal studies, I think, are a huge gap right now. They're really hard to do, but they're really, really important, I think, especially when we talk about mental illness and trying to predict risk and things like that, and getting a handle, again, on the within-subject variability and how that relates to the across-subject variability, sampling the same individual multiple times, getting baseline measurements on people who then go on to develop illness. This is really important, and again, there are some data sets out there now and some work on this, but I think this is a huge area that we need to be thinking about. Okay, and then we bring someone in, and what do? We have them in the scanner, what do we acquire? I've already talked a little bit about scan condition, this idea of a stress test. I've focused here on function since my title had fMRI in it, but obviously there's also a lot of meaningful anatomical differences, and I think the ideal approach would be to combine these in some way, structure, anatomical connectivity with DTI, function, etc., behaviors. I've talked about how do we get robust measures. A lot of these tend to be self-report, people filling out a psychological scale. I think the cognitive tasks and even the more naturalistic behavioral captures like free speech or things like this, or playing a game or social cooperation, some of these measures could be really, really interesting to try and predict from fMRI data. And yeah, I've already touched on this question of within versus between variability. Data analysis. So we have our data in hand. There's so many ways to analyze it, so many different flavors of even just functional connectivity analysis. I focused on the ones that I know best, but there's many, many other ways to do this. Bringing back activation, we can combine connectivity and activation changes. Again, some groups are working on this, but I think we tend to sort of pigeonhole ourselves into one analysis strategy. And especially using task-based data, the advantage with that is that you really can get both. So you can calculate connectivity during tasks the same way you can during rest, but you can also get these meaningful time-locked activation changes. And so what are the best ways to combine that to fully characterize an individual? One thing that I want to touch on also is coming back to this question of parcellation and parcel boundaries. So I've focused on sort of a plurality of the literature that tends to adopt a single atlas and kind of apply that to everybody equally, but there's a lot of evidence that individual specific parcellations could be really useful. So the idea there is that it's not, is that there's variability not just in the connections between regions, but also in the boundaries of the regions themselves. And so how do we capture that? There's been some really nice recent work looking at that. I think I've alluded to this data set, but this was the Midnight Scan Club data set where 10 people at WashU scanned themselves at midnight because that was when they could get the cheapest scanner time, and they did that over and over. I think there's at least 10 sessions of a few hours each for each person. And it turns out that with that much data per individual, you can start actually parcellating individual brains, and you can start to see differences between people that are very subtle but very, very stable. And so you can get the same types of boundaries across different days. And so that, of course, opens up the question of, are these meaningful? Can we use these boundaries to try to predict behavior, things like that? And Thomas Yo's group, again, had another very recent paper where they observed a similar thing. So with multiple scans from the same subjects, they could find these really fine-grained parcellations that were specific to individuals. And most exciting for me is that they also found that they were able to use the boundaries of these nodes to even start to predict behaviors. So by looking at, by bringing in a new subject and looking at which subject in the training set had the most similar, parcellation boundaries to that subject, they were able to get some traction in predicting some cognitive measures based on not the connections between regions, but just the boundaries of the regions themselves. So that's something we really need to be thinking about. Do we need to move, all of us need to move towards individual specific parcellations. And then finally, applications and interpretations. So individual differences are really interesting, but they can also be kind of a touchy area, and they bring up some interesting questions. So how mutable are these things? How changeable are we? How do they change with development, over the course of healthy development or aging? How do they change with disease progression? How do they change with plasticity and training? And then some of the applications, again, just ever pushing towards this, trying to come up with a tool that has translational utility based on fMRI. But I think there's also some interesting ethical questions that come up with this. So even if we had a test where we could perfectly predict, how someone's going to do in school or how they're going to respond to some intervention, I think we're going to need to think. And again, I think that's way down the line, but I think as a society, we need to be thinking about these things, and in a world with finite resources, how do we make sure that we're not stratifying people too early or things like this. As with anything, we want to make sure that we're doing this in a well thought out way and we're considering all the ethical implications. Okay, so that's bringing me to the end here. There's a lot of references in the talk, but these first three reviews are ones that have helped me a lot. There's a lot of literature out there, but these are good places to start if you want to learn more. And this is just a plug for my own recent sort of semi-review, semi-empirical paper. And then, again, many of you are probably aware of this, but there's a lot of open data sets out there now with both brain and behavioral measures, and these are pretty fun to play around with. You don't need to limit yourself to, doing an analysis just within one of these. If you have a data set in the lab and you want to, you've developed a marker and you want to see if it generalizes, there's increasing numbers of these data sets with behaviors that you might be interested in, so you can kind of combine results in interesting ways. With that, I'd like to acknowledge everyone here in my section, as well as everyone at Yale, which is where I did my PhD. You're welcome to email with any questions or comments, and I'd be happy to take any more questions now. ahead. Yeah.
Have you ever considered any dimensionality reduction tools like PCA, ICA, LDA?
Yes, so the question was, have we considered dimensionality reduction techniques like PCA or ICA, things like this? There's actually a really nice paper that just came out a couple weeks ago, somehow didn't make it into this, where they showed that they basically did a big group level PCA on functional connectivity matrices, and by reconstructing the individual matrices using those factors those components that were identified, they were able to get even better identification rates of subjects by sort of doing that dimensionality reduction first, and then reconstructing each subject as a function of those components. So I think that can buy you a lot in terms of increasing SNR, increasing interpretability. I think it's comforting that it sort of works on the more raw data, so to speak, but I think those approaches have a lot to add in terms of improving our numbers.
So can we integrate this with kind of group level analyses? So you showed that one figure that had, most of the, I guess it was from the midnight scan cut, maybe, where most of the variance was accounted for by individuals, but then the big purple frame was and this is what you get from the task. So, but do you think that there's a way to capitalize on this? Can you actually control for the variation and get a better sense of what is common?
Yeah, I think so for sure, and I think that's where, again, some of these dimensionality reduction techniques and using group level information to constrain what we're looking at individuals, absolutely. I mean, there's already some really great work out there that's doing that, and I think it's a question of, I see it as a balance, right? Because obviously there is something that's common to 99.9% of brains. We all have grossly the same macroscale anatomy. You can see grossly the same networks and things like this. And so I think it's a question of where do we want to be on that slider of certainly some level of group information is probably going to help us. We can sort of subtract it out maybe or see it as background that then, if we get rid of it, would sort of amplify the individual differences. And so, but if we go... Yeah, I'm not articulating this super well, but if we bias ourselves too much towards looking at deviations from what's common, there may be things that are not coming out in our group level analyses because there's so much variation in individuals. So it starts to be sort of this chicken and egg problem of, do you want to start by assuming everyone's the same and then look at what's different, or do you want to start by assuming just everyone's different? And, on the one hand, you have more noise, but on the other hand, you might be wiping out certain meaningful things, but overall, I think taking what we've learned from groups and coming up with meaningful ways to say, okay, this is what tends to be common, these are the deviations, which deviations are meaningful, that's how we need to be thinking.
And then I guess a related question, I'm trying to wrap my head around what it means that the tasks where people perform the most similarly are also where you can identify the differences the best. So do you think it's kind of because once you get people into a similar state, then what do you think they're driving?
Yeah, so, and I apologize if I wasn't clear about this. So it's not necessarily that they're performing similarly. So actually, the task that, at least in the Human Connectome project, and again, a lot of the in-scanner tasks tended to be fairly easy, so there's a lot of ceiling effects there, but the task that actually was most challenging, so we saw a lot of variability in the performance here, but everyone is still doing the same task.
Oh, okay.
So,
okay.
right.
There might be really different strategies, or when somebody's watching a movie, they might have different associations, but they're engaged.
Yeah, I keep trying to come up with a good analogy for this. I think it's just, you're driving the system in a way. You're not just, and you can think of it, one of the analogies that's getting a little reused now, but I think it's still very appropriate, is a cardiac stress test. You might not see heart problems when people are just walking around at rest, but all of a sudden you put them on a treadmill and make them actually use that system, and then they come out. And so I see that as sort of the analogy for this. And I'm fascinated by the differences within tasks. It sort of makes sense that giving any task, might be stabilizing these things, or at least just, making sure people are engaged and awake. It could be something as low level as that. But then, once you sort of start to see these differences between the tasks, and even in something like a movie where they might not even have an explicit task, but they're attending to this thing, what does it mean that we're sort of making everyone more similar, but we're still able to highlight these. I don't know. I mean, it's ongoing, but it's really interesting.
More of like a philosophical question. Why do you think it is important for us to know the individual differences?
Yeah, so the question was on a philosophical note, why is this important? I mean, I tried to make the case that just for practical reasons, if fMRI is ever going to be useful in the clinic, I think this is the direction we need to be going. And I see, I guess I'm Ardockian in the sense that I see looking at individual differences in healthy people as just sort of a stepping stone towards then extending that to people that are suffering from either at present or future neuropsychiatric illness. And so that's a practical reason, is to try to get a handle on the full spectrum of variability in humans, and if we can use that, in a medical context to improve how we diagnose and treat and whatnot. And then, I mean, there's also interest, at least on my end, from just a basic psychological cognitive perspective. We're all different. We're all special snowflakes. We all have our own strengths and weaknesses and idiosyncrasies. And, I think a lot of that, maybe all of that, is reflected biologically in our brains. And so just getting a handle on that and seeing what that looks like.
It's very difficult to use that clinically because it's very expensive. So I think that probably would be next step would be to go further and find out some clinical correlates that those are the differences, and then what we can use much cheaper than putting people in the scanner and seeing the difference. So did you think about differences like using EEG, which is much cheaper and can be used in doctors' offices as a correlation to the differences in resting state?
Yeah, absolutely. The burden is on us as fMRI researchers to show that whatever we're coming up with outperforms cheaper measures like behavior, like EEG. I guess my broad answer to that is we have decent tools for mental illness right now, but we don't have great ones. It's still very hard for us to predict, who is most at risk, who's going to respond to which treatment, whose illness is going to progress versus, who is going to get better on their own, things like that. And so I just see this as we're trying to fill some holes that definitely do exist, and you're absolutely right in that there's a financial burden associated with MRI, and if we can use these tools to sort of get a handle on what's going on and then find other ways to assess them that are cheaper, that would certainly be also a great outcome. But yeah, I think we can do better, and so that's, there's hope that fMRI can fill that gap.
By the way, I'm very impressed with your presentations.
Oh, thank you. All right. Thanks for coming on Friday.